Hi, we're getting near the end of the respiratory system, and I'd like to talk in this lecture about the transport of oxygen and carbon dioxide and the oxygen hemoglobin dissociation curve that we'll take a look at. So the first slide is pretty simple. It's just showing the transport of oxygen. It's done two ways. So one way that oxygen is transported is that a small percentage of it, less than 2% of it, just dissolves straight into the plasma. So here's the oxygen in the alveoli of the lungs. So this is representing lungs. Diffuses across the respiratory membrane into the blood. And about 2% of it just stays in the plasma. Not very much. It dissolves poorly in the plasma. Most of the oxygen enters the red blood cell, so greater than 98% of it enters the red blood cell and binds to hemoglobin. Oxygen plus hemoglobin. And this is referred to as oxyhemoglobin when it has oxygen bound to it. Oxyhemoglobin. And then that is transported to the tissues through the blood. So it goes to the cells, and then the oxygen comes off of the hemoglobin. Remember the partial pressure that we dealt with in the previous lecture in the tissues are about 40, and in the blood it's 100, so it pushes off out of the blood, goes down its gradient into the tissues. And we are able to deliver oxygen to the tissues where it's used for cellular respiration, back in Biology 160. And that's about it. Pretty simple. And then the red blood cells carrying the hemoglobin go back to the lungs, and they just pick up another load of oxygen on the heme group and transport it to the tissues, and goes round and round. So remember that the oxygen bound to the hemoglobin, remember that it's bound to the iron in the heme. That takes you back to the first lecture in the course, the first unit on blood. Okay, so that's the transport of oxygen. Pretty straightforward. A couple of things now to consider with oxygen is how readily it comes off of the hemoglobin. So this is just a standard curve that's constructed in the lab, but it shows the behavior of oxygen binding to hemoglobin. So across the horizontal axis is blood at different partial pressures. So in the alveoli, we've said that it's about 100, and off in the tissues, those are resting tissues, it's about 40. So that's just the concentration or the partial pressure of oxygen. Under those conditions, at those partial pressures, hemoglobin has more or less oxygen bound to it. So you just read the curve. For example, at 100 millimeters of mercury of oxygen, the hemoglobin is not quite 100%. It's pretty close. It's about 98% saturated. Approximately 98%. Okay. And the interesting thing to note on this curve is that it's relatively flat. At this point. So, for example, even at 60 millimeters of mercury, so if you drop by 40%, you go from 100 down to 60, how saturated is the hemoglobin? Well, you just read up till you hit the curve up here, and it's pretty close to 90%. So even though the partial pressure of oxygen has dropped from 100 to 60, hemoglobin is still approximately 90% saturated. Then, as the oxygen level, the partial pressure begins to drop further, this curve starts to get steeper. So it's flatter and steeper here. So at rest, we've dropped 60% now. We've gone from 100 to 40. And at 40 millimeters of mercury, how saturated is the hemoglobin? So this would be hemoglobin in your venous blood leaving the tissues. It dropped off oxygen, picked up carbon dioxide. How saturated is the hemoglobin? Well, it looks like it's pretty close to maybe 75% saturated. So 3/4 of the iron atoms in all the blood are carrying an O2 molecule. Okay, that's at rest. Now look what happens when you get further into the steeper part of the curve. Let's say that the cells are now exercising vigorously, and we're now down to 20. Well, at this point in the curve, I sort of drew over it, but it's about here. At this point, we're probably maybe a little bit less than 35%. 35% saturated. So it's given up quite a bit. We went from 40 to 20 in partial pressure, and the amount of oxygen that came off was significant. We went from 75 to about 35%. Which makes sense, right? Here the tissues are working hard, they need more oxygen. More oxygen comes off of the hemoglobin. Here we're at rest, lying on the couch, don't need as much oxygen in the cells, so a little bit comes off, still about 75% going back to the lungs to get some more. Once we get up and start moving and exercising vigorously, the cell demand for oxygen is much greater and much more oxygen comes off. Okay, so that's the basic oxygen hemoglobin dissociation curve. This slide, I realize, is a little hard to see, so I'll help you out. On the top figure is just the standard curve. So this is what we just looked at. It's actually showing volume, but I'm not going to worry about that. So let's just make this the standard curve. Okay, we just talked about that in previous slide. So I probably could have cut that off and enlarged these two. But I think if I help you out here, it'll still be okay. This set of curves we still have across the bottom in both cases is the partial pressure of oxygen in millimeters of mercury. And on the vertical is the percent saturation. Percent sat. Getting a little messy here, but percent saturation, just as before. I know you can't really read that. I can't either. Okay, so the middle and the bottom panel. The middle panel is showing changes in pH. How does pH affect the standard, the dissociation of oxygen? So you should be able to see a blue line on the bottom, pink line in the middle, gold line on top. Okay. So the middle line is representing normal pH. So the pH is 7.4, halfway between 7.35, 7.45. So this is normal. All right, and then you should be able to see that the blue line is below that. So the blue line is representing pH at 7.2. The blood is acidotic. So we have acidosis. The other line at the top, the gold line, put a little green dot, that line is pH 7.6. So here it's alkaline, it's basic, so this would be alkalosis. Okay, all right, so how do you interpret this? How does the oxygen affinity for hemoglobin change with pH? And we're talking about the curve either shifting to the right or shifting to the left. So the blue line has shifted right. Shifted right, and the green line has shifted to the left of normal. Shifted left. So which one of those has a higher affinity? Under which condition will oxygen come off of the hemoglobin? Well, let's look at that where we have a little bit more room to kind of get the idea on how to look at that. So we'll just draw a set of axes, and this will be our partial pressure of O2 millimeters of mercury. We won't put numbers on it, it doesn't matter. And this is percent saturation. Okay, so what does it look like? It looks sort of something like this. So let's call this one normal. And this is normal pH. And then let's just, for comparison, look at the blue line for acidosis. Well, it shifted to the right. So draw the line maybe sort of something like that. So this is low pH. All right, so how does that affect the oxygen binding? Well, to kind of get your mind wrapped around it, start with any point. So let's say that at this partial pressure here, how saturated is the hemoglobin? Well, you just go straight up from here. And at this point, you read across and hit it there. And the other line, the normal, is here. So what happened to the oxygen bound to hemoglobin when the pH dropped? Does more oxygen come off or less oxygen come off of the hemoglobin at low pH? Well, clearly quite a bit more comes off the way I drew it. Normal pH was at a higher percent saturation. At a lower pH, there is less oxygen bound to the hemoglobin. Now, does that make sense? So if the tissues are exercising vigorously, they can be producing lactic acid if they have to metabolize anaerobically. There are other metabolic acids. So exercising skeletal muscle, pH drops. Well, this facilitates more oxygen being delivered to the tissues. And just the opposite when the pH is increased and tissues become alkalotic. So if I can clutter up this drawing a little bit more and put a different curve on and say this is a high pH, you can just kind of get the idea by looking at the intersection on the axis. So even less oxygen comes off of hemoglobin. So in some ways this is sort of physical characteristic of the hemoglobin in response to pH. So main point to note is that when the curve shifts to the right, it promotes dissociation. So here the blue line, low pH, shifted to the right, away from the normal, and that facilitated oxygen dissociating from the hemoglobin and delivered to the tissues. The bottom figure is pretty much the same idea. The curve is shifting right or left. What you can't see are the numbers, so I'll help you out on that. So this line, this is temperature. So that blue line is 10 degrees C. Cold fingers, cold toes. The next line is at 20 degrees C. Chilly fingers and toes. That's not your core temperature, otherwise you'd be dead. Severely hypothermic. And this is, I believe that's 37. It might say 38. I'm going to call it 37 degrees C. This equals 98.6 degrees Fahrenheit. So this is normal. And bottom curve is at 43 degrees. Feverish. So again, the idea is to note how the oxygen and hemoglobin dissociate from each other as temperature changes. So again, the curve is shifting either to the left or to the right from normal. And does that promote or inhibit oxygen hemoglobin dissociation? Okay, continuing on with the same idea, here are two different hemoglobins. One is produced during fetal development in utero. We talked about this earlier in the blood lecture. And then we have the adult hemoglobin. And after birth, the fetal hemoglobin is broken down and adult genes turn on and produce adult hemoglobin. So, in utero, remember that the fetus needs to get all of its oxygen stolen from mom's blood at the placenta. So here are two curves, and if you're doing black and white, it's a little hard to see with the color, so I'll go ahead and label this. This is the adult hemoglobin curve, and this is the fetal hemoglobin curve. So which one has a higher affinity for oxygen? Well, we could just take what they started with, Start at, let's say, 26.8. Doesn't matter. And you just read up. They've done it one way. You can do it another way. This is how I kind of prefer to do it. You can look at it either by 50% or by the partial pressure. I think this might be easier. So ignore the other line for right now. Just go with the 26.8. Doesn't matter. We could have done 40. We could have done 30. But you just go straight up. So at 26.8, adult hemoglobin is 50% saturated. At the same partial pressure of oxygen, the fetal hemoglobin is a good deal more saturated. I don't know, 50, 100, let's call that, I don't know, let's say maybe 70%. So the fetal hemoglobin has a higher affinity. It'll steal, it'll grab, it'll bind onto the oxygen tighter than adult hemoglobin, which makes sense because the fetal hemoglobin has to go by maternal hemoglobin and the oxygen needs to be transferred. So the last figure, the last slide in this lecture is on the transport of carbon dioxide. So we started with the transport of oxygen. That was pretty straightforward. And then we spent several slides looking at the oxygen hemoglobin dissociation curves and different parameters that affect that binding. The final slide in this lecture is the transport of carbon dioxide. And that's done three ways. Remember the two ways for oxygen? About 2% dissolved in the plasma, 98% carried on the hemoglobin, in the heme. Carbon dioxide is transported three ways. Some of that dissolves easily in the plasma. And now we have about 7%. So that's one way. 7% of the carbon dioxide just dissolves in the watery plasma. About 23% of the carbon dioxide that's transported binds to hemoglobin, but it does not bind to the heme. It binds to amino acids, which I'll abbreviate as, well, let's just spell it out. I was going to say AA. Amino acids on the globin portion of the hemoglobin. And about 70% of that carbon dioxide, the vast majority of it, is converted to the bicarbonate ion. So that we need to go back and review. So just to work our way through the slide, here we are in the tissues, the feet, fingers, wherever, and we're generating carbon dioxide from cellular respiration. Perhaps you remember some of those steps of the Krebs cycle. So there's all the CO2 that's being produced in the tissues and goes into the blood, and about 7% of it is taken care of, just dissolves straight into the blood, into the water of the plasma. No chemical change. The remaining 93% enters the red blood cells. And of that, about 23% of it binds to the hemoglobin. And we have HbCO2. And this is referred to sometimes as carb-amino-hemoglobin. And we've loaded now about 30% of the carbon dioxide. We've accounted for about 70%. The remaining 30%. The remaining 70% binds to water, and here's our familiar reaction that we've done before. Carbon dioxide plus water. The enzyme Ca stands for what? Carbonic anhydrase. Very good. And that produces this. And this is what? You need to remember this reaction. So there's our carbonic acid. And the acid dissociates. A hydrogen comes off. There's the hydrogen. And that's going to cause the pH to drop, unless we bind it to a buffer. And when we talked about blood buffers, we talked about proteins doing most of the buffering. So the hydrogen binds to the hemoglobin. Remember, we're at the tissues, so the hemoglobin has lost its oxygen. This would be deoxyhemoglobin. Binds to the hydrogen, and then we go back. We're done with that. We go back to the lungs and get rid of it. The bicarbonate now, check this out. The bicarbonate ion that was produced leaves the red blood cell. So it leaves the red blood cell, goes out into the plasma, again, as bicarbonate. So the bicarbonate was made in the red blood cell, it goes out into the plasma. But as that negatively charged ion leaves the red blood cell, another negatively charged ion, chloride, enters. And this is referred to as the chloride shift. So we're at the tissues. Bicarbonate ion is produced, it leaves the red blood cell, and is replaced by an incoming chloride ion. Keeps the charge inside the red blood cell more or less constant. So we've loaded the carbon dioxide at the tissues, and now we go off to the lungs. Gets transported to the lungs, and now everything runs exactly backwards. All you have to do is turn all of the arrowheads around and move them in exactly the opposite direction, and we're done. So all of this upper part of the story has now been delivered to the lungs, and we need to get rid of the carbon dioxide. Well, the 7% that was dissolved just goes straight out into the lungs. And the carbon dioxide that was on the hemoglobin, or carb-aminohemoglobin, separates. That carbon dioxide then leaves. And we're left with the hemoglobin, which will pick up oxygen because we're back at the lungs. So it transports both carbon dioxide and oxygen, different spots on the molecule though. And to deal with the bulk of it, 70% of the CO2 that was converted to bicarbonate, right? Here's a CO3. Well, the CO2 is in there. So the bicarbonate ion enters the cell again, and now we have the chloride shift goes backwards, just opposite. Turn the arrow around. The chloride comes out, so there's our chloride shift at the lungs. And then here comes the bicarbonate, and you know the reaction. It combines with a hydrogen, which came off of the hemoglobin again. And we're back to, what's this again? Good, carbonic acid. The enzyme carbonic anhydrase splits it back to CO2 and water, and the carbon dioxide leaves also. So you should be able to follow along with that. I would practice this. I would draw it. Once you spend a little bit of time on it, it's pretty straightforward, I think. So I'm going to encourage you to practice. It may look busy, but remember these 3 ways. This is what you need to memorize first, are the 3 methods that carbon dioxide is transported. 7% in the plasma, 23 on the hemoglobin, and 70% bicarbonate, and then you should be able to figure out the rest of it. Okay, good luck. I hope you have some questions, and do spend some time practicing this. And we'll see you for just a couple more short lectures soon. Bye.